So anyway, welcome everybody. So my name is Matt Anderson. I'm the Vice President Medical Director at Blood Center of Wisconsin. I'm also going to be serving as your moderator for the session this afternoon. I'm also proud to say I'm a BioForward board member, and I know it's been at least personally exciting for me to see all the passion amongst this group here for the future of biohealth here in the state of Wisconsin. So I'm excited that we have a great panel for you this afternoon. So we have Dr. Broeckel, who's the CEO of RPRD Diagnostics, Dr. Ames, who's an application scientist at Biomax Informatics, Dr. Capitini, who's a medical oncologist and assistant professor of pediatrics at UW School of Medicine, and then Dr. Wisinski, who's a medical oncologist at UW Carbone Cancer Center. And just a little bit, I'm going to have them come up and tell you a little bit more about the work they're doing in the precision medicine space, and then we'll follow that up with a bit of a discussion session. But to kick things off, I was going to at least do a quick introduction of precision medicine, talk a little bit about this paradigm and what the impact might be in terms of healthcare and industry, not only in our state, but beyond. So I always like to start with patients. I'm a physician myself, and I think these two patients illustrate where we're doing well in terms of precision medicine and where some of the challenges may lie. So on the left here, we have a patient who has breast cancer, and as many of you are well aware, over the past 2 decades, we've learned a lot about the molecular basis of breast cancer, and we've been able to use that information to not only better diagnose these patients, but also do better prognostication, and also develop targeted therapies that can target the molecular aberrations in the tumor. On the right here, we have a patient with type 2 diabetes, and as we know, this is a huge burden to the US healthcare system. But despite that, we don't have a lot of molecular information that helps us predict who will develop type 2 diabetes, and then when they do develop diabetes, where could we intervene on a molecular basis for these patients? So again, an example of an area where we're doing much better in terms of precision solutions today and where we need to do better in the future. So in terms of just setting the stage of a definition, and I'll freely admit this is kind of my definition, but loosely thinking about using molecular information defined very broadly to provide precise diagnosis and treatment for an individual patient. I think this is what most of us think about when we think about precision medicine. But it's also about defining groups of patients that will or will not benefit from a particular therapy, for example. But also thinking about the practice of medicine. So how can we reclassify disease categories based on the underlying molecular pathogenesis? And through this, we're really moving from a patient to a class of patients to an entire system of medicine. And this broader concept of precision medicine is what really been driving some of these larger research initiatives, such as the Precision Medicine Initiative that was started in the last years of the Obama administration. And precision medicine really started with genomics because that was one of the first technologies that was available to us. And actually, one of the earliest examples of using genomics to affect patient care actually happened right here in the state of Wisconsin. You may or may not know about this story. This is actually Nick Volker. So as a child, Nick had extremely horrible inflammatory bowel disease, which is very unusual for a child that age. And under a research protocol, investigators at the Medical College of Wisconsin sequenced Nick's exome and actually found a mutation in a gene that essentially puts a brake on the immune system. So Nick's immune system couldn't stop once it got activated. And really the only treatment for this was to have a hematopoietic stem cell transplant, which again is not typical therapy you might imagine for inflammatory bowel disease. So folks at the Children's Hospital of Wisconsin actually did the stem cell transplant for Nick, and he is doing much better today, having a much more normal childhood, is able to go to school with other kids. It's a really powerful story, and in fact, this was actually written up in a book by 2 reporters at the Milwaukee Journal-Sentinel, so I recommend you check that out if you're interested in that story. And our ability to do better DNA sequencing and interrogate genomic information has also been applied to cancer, and it's led to some interesting insights. Here's just one example. Here's an example of 2 diseases that currently we think about in current medical practice as being very distinct and different. So on the left, this is a relatively rare tumor called a gastrointestinal stromal tumor. These patients usually present with a mass in the belly and some abdominal pain, and these are what the tumor cells look like under the microscope slide. On the right here, we have a disease called systemic mastocytosis. These patients present with skin rash and ischias. And under the microscope, you can see here the tumor cells that are kind of percolating throughout the skin. And when you look at this from a kind of phenotypic perspective, these patients are typically seen in the hospital in two very different settings, one in gastroenterology and surgery, one in dermatology. But if you understand these diseases at a molecular level, it turns out they share something in common, which is actually the gene that drives these processes, and this is the c-KIT gene. So you can imagine a day where these patients are not necessarily treated by where the disease symptomology is, but the genetic underpinnings of their disease. And a lot of the advances in precision medicine have come in the cancer area, and here's an example. I think everybody can do this diagnosis. This is a malignant melanoma over here. It turns out that many cases of malignant melanoma harbor a mutation in the BRAF gene. This is the V600E mutation. And once that was discovered, pharmaceutical companies could use that to actually specifically target these cancers. And here's just an example from a clinical trial where an oral agent against this BRAF inhibitor was used. This is a patient who has widely metastatic melanoma, which is essentially a uniformly fatal diagnosis. After being on the drug, you can see these tumors have essentially melted away. And what's interesting is as we study more cancers, we find that other cancers also carry this BRAF V600E mutation, so this drug can also be used in different settings. So here's just some examples, such as ovarian cancer and salivary duct carcinoma, that you can actually use these BRAF inhibitors and see clinical efficacy. So the problem is, though, as I alluded to in the first slide with our two patients, that we don't have all the answers yet for these very complex multigenic diseases such as diabetes. And just to illustrate that, I want to show you the case of the healthy Stanford professor. So this is Dr. Steve Quake, and Steve's a professor of bioengineering at Stanford University. A real smart guy, and he actually created his own sequencing machine at one point. So typically inventors, when you make your own machine, what are you going to do? He's going to sequence himself. But before he did that, he actually was smart and met with some genetic counselor just to prepare himself for what he might learn in his own genome. One of the things that Steve was particularly concerned about is he had a relative that died at a pretty early age, 19 years of sudden cardiac death. So naturally, Steve was worried if his genome had any risk alleles for sudden cardiac death. But in addition, if you look at his family history, there's common diseases. Many of these we probably all have lurking in our family history in this room, such as coronary artery disease, osteoarthritis, congestive heart failure. Nothing too unusual or strange there. And after he did his genome sequencing, he partnered with another group at Stanford to essentially interpret his genome. And he asked the question, well, what does my genome tell me about my risk factors for various diseases? Well, the first good piece of news is it didn't apparently have any variants that predisposed to sudden cardiac death, so that was good news. And then he asked, well, what about the risks of other diseases? And these diseases are shown here in the larger type. So coronary artery disease, obesity, osteoarthritis, type 2 diabetes. Well, I mean, I guess I would make the argument that if you just simply looked at Steve's family history, you might have already predicted that he would have an increased risk for these diseases. So then the question became, well, now that he knows that he has increased risk for these diseases, what can he actually do about it? And all that is located on the outside of the circle. These are probably words very familiar to anybody who's recently seen their family practice physician. Diet, exercise, stress, don't smoke, and drink alcohol in moderation. Again, I may make the argument that you don't necessarily need a genome to tell you to live healthily. And so that's really where big data starts to come into play for precision medicine, especially these complex multigenic diseases. So just to illustrate this graphically, so we all live in different places in phenotype space. We all have different states of health and disease. And in addition to that, we already know that we have different genomes. We also have different ways in those genomes are regulated, different ways in which those genomes are transcribed or expressed, different bacteria and viruses that live both on us and in us, and different things that we've been exposed to throughout our life. And it's really the integration of where we live in all of these various planes that actually paints the most accurate picture of where we are at any one point in time with our health and our disease. And that's a lot of data to integrate and put together. And we've moved on. I mean, as I mentioned in the beginning, we started kind of with genomics, but precision medicine is much more than genomics. So as we're seeing more recently, we're harnessing the power of the immune system to actually attack cancer cells. We'll hear more about that today. Looking at other complex biological molecules in the body, such as the proteome, and then using our stem cells for regenerative medicine. That's another area of opportunity that's also a precision medicine approach. I mean, the good news is there's a lot going on in this state. Here's an example just in terms of kind of the academic approaches. There's a group out of Marshfield Clinic that has a grant through the Precision Medicine Initiative together with these partners to enroll patients. Our healthcare partners are, especially in the molecular aspect of cancer, having molecular tumor boards where they're matching patients' tumors to particular therapies. And we have a lot of companies and industry that are active in this space, not only companies that do direct patient diagnostics, but groups that are involved in cellular therapy, and also companies that create the tools and technology that we use to further these precision medicine approaches. So with that, I'm going to turn over the presentation to Dr. Wisinski, who's going to tell you a little bit more about the work that she's been doing. Dr. Wisinski?
Hi, I'm Kari Wisinski, and I am a medical oncologist at the UW Carbone Cancer Center here, and also a clinical trialist, meaning that I think a lot about how do we ask clinical research questions studying human patients to understand whether different approaches work for that cancer. So in particular today, what I'm going to talk about the NCI MATCH study, which is a precision genomic study in cancer patients. So thinking back, right, we used to classify cancers, and we still do to some extent, really by where the cancer starts in the body. Whether it's a breast cancer, a colon cancer, prostate cancer, where does that cancer start? But really, we know that that is a very non-exciting, non-specific way of thinking about cancer. It's very generic. And we've learned a lot about cancers in the last few decades. We were just talking a little bit about an example of breast cancer, where we've learned a lot about which breast cancers are driven by estrogen and which ones are driven by HER2, a protein that can be expressed in a portion of breast cancers. But really, we know that it is much deeper than that. So here is a molecular profiling assay that's been done on breast cancers. And breast cancers can be split into some subtypes based on estrogen, progesterone, and HER2 expression. But even in each of those subsets, you can see here, just a picture of it, right, that there's a lot of heterogeneity in terms of the genes that are altered in that cancer. And some of those genes are likely driving the cancer's growth. This has also been seen in lung cancers and many other types of cancers. So how do we answer the question if we have common cancers that are really multiple different disease types, and even in those disease types, multiple different subtypes, how do we answer the question of whether a targeted therapy against a specific genomic abnormality works or not? And it's really important to answer this in the context of a clinical trial. So really, when we think about what we're looking at is that we can detect a genomic alteration, right? We can have a test that can identify a genomic alteration, but that doesn't always tell us for sure, doesn't really predict whether that is a driver of the tumor growth and whether a specific drug targeting that alteration is actually going to have any effect for that individual. A second challenge is that resistance can also emerge during a treatment. So maybe at a baseline, a specific genomic alteration will predict that a medication will benefit that individual, but ultimately resistance can occur. And so clinical trials allow us to ask these questions. Does a medication work? And when it stops working, or if it stops working, what are those pathways or mechanisms that start to emerge? So one example of this is, I was alluded to a little bit before. So there is this BRAF mutation that's been well defined in melanomas, and there's a BRAF inhibitor called vemurafenib that's been shown to have significant benefit in melanomas that have the BRAF mutation. So about 50% of patients will have a dramatic shrinkage in their tumor that occurs with treatment with vemurafenib, and that was established in clinical trials. But also, we know that that doesn't always predict in other tumor types. So here's a study in colorectal cancer. Same drug, same BRAF mutation. Everybody thought that this would be clear-cut, right? That should result in a 50% response rate. But really, the response rate was pretty abysmal. About 5% of the patients in these studies benefited. And we've subsequently learned that's because a lot of colorectal cancers harbor a second mutation that leads to de novo resistance to these agents, and that potentially combination therapies would be more effective. And even in melanoma, where we saw dramatic responses to BRAF inhibitors, the challenge of acquired resistance definitely emerges. So about 6 months after starting therapy, even patients who have had a really dramatic response, many will start to have resistance emerge and tumor progression develop. And so there's been a lot of analysis of what's happening at that time in the tumor, what's actually emerging on a tumor level. And what's been found is that a second pathway starts to become altered in those tumors, called the MAP kinase pathway. And that there is a medication called trametinib that targets downstream in that pathway, and when combined with vemurafenib, improves the duration of response to therapy. So that set the foundation for these big, large national studies that are going on. And I'm going to talk about the MATCH study, which I'm involved in nationally. So MATCH is the Molecular Analysis for Therapy Choice Study. And MATCH was really designed to allow us to have an infrastructure to ask the question of how genomics is going to help direct treatment for individual tumors, especially when we talk about this heterogeneity that I showed you earlier on, right? If there's a molecular alteration that's only present in 2% of estrogen receptor positive breast cancers, it'll be nearly impossible for us to do a study to look at that 2%. It's just too small of a cohort. And maybe that 2% of breast cancers, maybe the same person who has the same alteration in a lung cancer might have a benefit too. So instead of separately studying them, let's merge them together and ask the research question in a basket study. So this way the MATCH study is designed is that 6,000 patients with advanced cancers that were solid tumors or lymphomas or multiple myeloma, for whom there was really no standard treatment that was a part of their care that was recommended, had their tumor biopsied at the time that they enrolled in the study. So this means it was a reflection of a recent alteration of tumor. The patients then had the tumor sent for sequencing, genomic sequencing, and it was identified whether they had an actionable genetic alteration, and then targeted therapies were administered to the individuals based on their alteration. And there were 24 to 30 different small studies in this large basket study. So the key eligibility criteria were kind of standard. So again, certain cancer types that we talked about, no standard treatment left, and no other serious medical conditions that would limit being able to participate in a study. And here's the genomic platform that was used. This was an AmpliSeq and Torrent platform, looking at 143 genes and over 4,000 actionable mutations of interest. And then there was also some IHC analysis that was done. And this was done at 4 sites that had validation across the US. This is the 24 original arms, and I just wanted to point out the arms that I was involved in was Arm B and S1 here. And again, these are all rare alterations in tumors for the most part, although some are more common. And the drugs targeting them sometimes were similar medications used in different arms, but targeted that specific molecular alteration. So each arm had about 35 to 70 patients per cohort, and really what we were looking at is response. So again, patients who had the tumor biopsied, identified whether they had an actionable mutation, were assigned to a study drug, and then identified whether their tumors that were evaluable on imaging were either stable or responding or progressing. This is a standard clinical trial design. And then either continued on study drug, or if the tumor progressed, came off study, or if they had a second alteration, were able to continue on with a new agent. So there's some specific strengths of NCI match, in part the platform that's used for the genomic testing. The actionable mutations were well vetted by national experts to really try to identify those that were most likely to show benefit for these specific drugs. And there was a large collaboration with industry partners to be able to do this large study. And then incorporated in the study are analysis for mechanisms of resistance, so that if patients do benefit and then subsequently progress, there's another biopsy to look for some of these mechanisms of resistance. So MATCH opened in 2016 with 10 treatment arms, and then subsequently expanded up to 24 and now 30 treatment arms. It was open broadly nationally, and it met its goal of 6,000 patients in May of 2017. And currently, it's closed to some of the more common mutations because those cohorts filled, But some of those rare mutation cohorts really had very hard time finding enough patients to fill. And so now it's open to studies who have standard of care assays done in the clinic and can be enrolled and matched based on those findings. So lastly, that's about match, but I just wanted to have one slide talk about our molecular tumor board that we have here in the state of Wisconsin. This is run through oncology practices throughout the state of Wisconsin. And really what this is doing is just looking at standard of care assays that we might get from any source. And then there's a panel of experts, geneticists, pathologists, medical oncologists, you name it, all around a table that kind of discuss the results of that analysis, but also in the context of the patient's tumor and their other medical conditions. And then we try to make recommendations for clinical trials or off-label treatment, and then capture in a registry to figure out what happens to those individuals. So that every time we're treating a patient in the clinic, we're also learning for our next set of patients. Because as the testing has advanced, we need to continue to learn how we continue to treat each individual's cancer better. So, thank you.
Right, good afternoon. My name is Ulrich Broeckel, and I'm a co-founder and the CEO of RPRD Diagnostics. If I can get the slides. Here we go. And I'm also a professor at the Medical College of Wisconsin in the Department of Pediatrics. So I want to tell you a little bit about pharmacogenetics and why we think pharmacogenetics is really one of the key steps in precision medicine. And so in my academic life, we've been pursuing pharmacogenetics for over 10 years now, and really implementing the pharmacogenetic testing in clinical care. And so if you think about pharmacogenetic testing, this is really the response to patients to any particular drug which on the market is certainly variable. Not every patient, if pharmaceutical companies would like to tell you that every drug works for every patient, but it's actually not the case. The variation to a drug, to any drug, is variable, and it affects both the efficacy, also the occurrence of side effects and the toxicity, also the therapeutic window, and potential interactions if you look at patients who receive different drugs. And so for many indications, drugs from various drug classes are also available and make then the whole decision in terms of what type of drug you would like to pick for a patient often guesswork. And I think one of the ways how we can improve and really provide better care is by using pharmacogenetic testing and pharmacogenetics to find the best drug for a patient for a particular disease. So understanding and then very proactively managing the genetic information and finding the right drug for the right patient will really improve the therapy. And there are examples, multiple drugs published in the scientific literature. We can improve, obviously, patient satisfaction. There's nothing more than if you try a number of different drugs and none of these drugs work, and you have to try 3 or 4 until you finally end up with the right drug. So optimizing the treatment right from the beginning, I think, really improves patient satisfaction. It obviously also improves safety. If you think about toxicity, reducing toxic events, I think, will really improve that. And also, at the end, it will actually reduce healthcare cost and healthcare risk. So really, we also started out thinking about pharmacogenetics almost like 10 years ago to really implement this in patient care. That was a time when originally everybody thought, well, we're going to do genetic medicine, genomic medicine, and now we're in precision medicine as we extend more from DNA to really many, many more markers. And so we also thought that pharmacogenetics is one of those tools and methodologies you can develop to really build the basis to implement not only genetics, but all the other omics technologies into patient care. Because obviously not just measuring something or genetic testing, that's really just the very first step. There's a whole other environment which one needs to develop to really implement this. Now, thinking about pharmacogenetics, just to tell you a little bit about how prevalent this could be, but certainly is not, is if we think about all the drugs which are currently approved by the FDA, those are about 1,200 drugs, and about 7% of these drugs actually have actionable pharmacogene information associated with that. That's one way to look at this. You can also look at this just in terms of the prevalence of all prescriptions, because not every drug of these 1,200 is prescribed in equal frequency. So you can look at the number of prescriptions and how often a genetic test potentially should be utilized. And there are about 4 billion scripts a year written in the US, and about 18% of all these drugs are affected or are linked to actionable pharmacogenes. Now, if I ask an audience, and I can ask you here, how often do you think physicians actually order pharmacogenetic tests? That's certainly not the 18% of these all actionable drugs. And one of the reasons is that pharmacogenetics, although it's been around for a while, is actually pretty complicated to understand, pretty complicated to implement. Physicians might not know where to order a test, and if they order a test, they might not actually know how to interpret this. So those are some of the hurdles which we experienced early on with pharmacogenetics, and as we were talking about extending this now to precision medicine, those are probably some of the same questions and some of the same problems we will experience moving precision medicine into this. Now, if we think about different diseases, so is pharmacogenetics really relevant to any particular disease, or is it just targeted? These are just some of the examples here in our disease spectrum. So it really covers all different diseases. So you don't have to be just a psychiatrist or oncologist to really understand that. We know a lot in oncology. There's genetic testing should be done or is actually currently done, but also psychiatry, neurology, epilepsy is a typical example where pharmacogenetics has been shown to really improve treatment and the selection of particular drugs, but also cardiovascular disease. If you watch the commercials for Plavix, it's an anticoagulant used in patients after a heart attack. Pharmacogenetic testing is actually recommended by the FDA, currently not really widely utilized. But also for infectious disease, GI, and pain, another area around codeine and morphine, where pharmacogenetic testing and the pharmacogenetic genes, relevant genes, actually play a very significant role. So if we're thinking about all these different disease entities, every patient should be covered by this. Every disease entity will be covered by that. And so our approach really, and I'll show you, I talked a little bit about one example where we work with one particular hospital, now we're extending this, is if this is so relevant here, why wouldn't we just provide genetic testing, very broadly based, test all relevant genes, at the time when a patient has a very high likelihood that in the future he or she will need any of these additional drugs or these drugs in these disease entities. And so that's really the concept around preemptive pharmacogenetic genotyping, where you genotype a patient ahead of time, you have the genetic information available, and then you can utilize this genetic information at the time when you would like to order a particular test. And so the concept of preemptive testing, this is cost effective because technologies are now available which allow you to very cost effectively analyze a broad spectrum of genes. And another really important thing for physicians is the turnaround time. So if you want to make a decision on what type of drug you would like to prescribe, if you order a test and you have to wait for a week or so, sometimes you just don't have the time. If you have preemptive testing and you have the results available, your turnaround time is basically zero because you have the information right available. So how does this now work in the overall context of delivering healthcare? If we have a physician who orders the test, we perform the genotyping, the data translation, the interpretation then is done at the lab, and a report is generated. And then this report, and this is really where technology comes into play again, gets reported back into a medical record. And I think one of the key things, what also the key requirements, I think as knowledge and complexity grows, integration into medical records and the decision support inside a medical record is an absolutely critical component in this overall circle here. Once you have that, then physicians are really enabled to utilize this information, whether it's now genetics or any of other precision technology methodologies. Here's just one example. I'll show you to demonstrate what I mean by pharmacogenetics is actually complex. So this is one gene, CYP2D6. A lot of drugs are actually metabolized in the liver and are affected by this gene. Here's just looking at about 800 patients here. We're looking at the complexity of all the different combinations you can have. And you can't ask any physician to really memorize, what this means if you have that result, and you translate this to what type of drug you would like to prescribe or should prescribe to your patient. It's not only the genetic result that actually gets translated into the activity of this particular enzyme, and then to add even more to this complexity is that sometimes if you're a poor metabolizer, that basically means this gene doesn't even exist for you. Sometimes that's something good, and sometimes that basically means you should not actually take this drug at all. So all these complexities we addressed, and we really learned and implemented this in a study which we did together with St. Jude's Children's Hospital in Memphis. It's the PGen for Kids study. Every child who currently is seen and is treated at St. Jude's has been preemptively genotyped in our lab. Those are now 4,500 children. All these results, the genetic results, are available for the physician, and each time a physician wants to add a new drug or prescribe a new drug, this information is compared against the genetic information, and interpretation and support is provided by this. So here's a publication describing our overall approach. This really requires automatic decision support inside the medical record. And this is a screenshot of the medical record, which is Cerner, not Epic. St. Jude's is on Cerner. And we also went so far as to automate the actual reporting of the clinical report. So there are different components which then, based on the actual genetic result, are provided to the physician. This is all automated. So automation and decision support inside the EMR is developed so that physicians can really get this information in a way they need it at the time when they need it to make the decision when they want to prescribe a drug. So in summary, we strongly believe that pharmacogenetics as an example of precision medicine and using this in a preemptive manner offers clinically relevant and important advantages to the implementation of pharmacogenetics in clinical care. For pharmacogenetics and genetics, and even more so for all the future precision medicine applications, what will be developed over the next few years, the integration into the medical record information system and decision support inside an EMR is absolutely critical. I don't think we can expect from any of our colleagues in the future, with all the tests what we're going to develop and all the information which is going to come out, that we're going to be expert in these areas. And so I think there's going to be a big role for precision medicine in the IT space. There's going to be a continuous ongoing clinical evaluation of our ability to demonstrate clinical value, and we're offering this as a testament for the implementation. So with that, thank you very much, and we will then answer questions at the end of the presentations. Thank you.
Good afternoon, it's a pleasure to be here. My name is Mercedes Ames, and I represent Biomax Informatics. We are a German company based in Munich, and we have a branch here in Wisconsin, and we specialize in data management solutions and data integration and knowledge management solutions. We are a multidisciplinary team of scientists, experts in different areas of the biological sciences and also computer sciences, bioinformaticians, mathematicians. And we work in different areas of the biological sciences, and of course, we have done work on health informatics. I think the understanding goal of health informatics basically is to provide the right treatment for the right patients at the right time and for the best therapy with a high efficiency, both in the quality of life of the patient or the good outcomes, but also in terms, in economic terms for the health system. So ideally, the vision of healthcare should be to enable every doctor, researchers, and patient to benefit from the experience and knowledge. And where do we accumulate experience and knowledge? In a practical way in the data we gather from them. So this requires a good use of digitalization. In many cases, just the use of the digitalization. There are countries in Europe that haven't done any digitalization of the medical records. So the healthcare step number one, from our point of view, is to fully have digital data and an information management system in place. The way we suggest doing it, and we have done it with partners in the European Union, is through the use of the semantic integration of different content types of data, a heterogeneous data, pretty much, and that's the healthcare step number two. And finally, is to use the information that we gather from that data and the experience to provide a solution. So this requires the use of different types of integration, data integration and knowledge management techniques. We use a semantic integration model that basically is a very intuitive model that relies on the biological question that is in place. And that allows us to integrate different sets of data, for instance, imaging data, clinical records, experimental data, databases, both free and open databases, and also some private databases, if we have access to, and the use of ontologies. The semantic integration occurs at the middle level. That's what we know, the knowledge model that allows us to gather, in this case, information, different types of information from the patient. And then a third layer that consists on querying and analyzing that data. This system allows us to have a very optimal management of the system without exhausting it. And also, the results help us to continually improve the model. And the advantage of using this kind of modeling for integrating data is that it offers the flexibility to add continuously different types of data and continuously add data to the existing objects that have been defined in the model. So it's a very dynamic model that allows you to add data without having to redesign the structure of your informatics structure. So in a practical case, for instance, we will have a model where we have a patient, and we will have different types of data integrated in the semantic model. For instance, omics data from genomics, proteomics, metabolomics, phenotypic, clinical, and environmental data that eventually will be having an impact on the outcome. And we can integrate also analytical tools, simulations, prediction analysis, basically machine learning techniques that allow us to either classify and predict a possible outcome based on the data we have fed to the system. So I am going to share basically a snapshot of a partnership that Biomax Informatics has with the Center of Excellence for the Treatment of Chronic Failure Diseases in the Netherlands. CIRO, the Center of Expertise, have multiple sets of data, at least 6,000 data points for each patient. The main problem they were experiencing is that, and this is basically a reality, that a patient cannot be treated the exact same way as any other patient that came before. However, we can learn from the whole group what could be the best way of treatment for a particular patient. So that was the conceptual idea that translates basically in gathering, then doing a baseline assessment of the patient and all their, all the elements that are part of the life, from the environmental conditions to the physical to the clinical aspects of a patient, make a determination based on clustering. I will show you in the next slide. Propose a treatment plan based on where this patient was classified, and then make an assessment on how the outcome came and also analyze it in the group context. As I mentioned before, each patient had a minimum of 6,000 data points. It included even, also genetic profiling tests. And it was conducted during 6 years, so the data accumulated during 6 years was used for building the clustering on the treatment established for a patient. For instance, if a new patient comes to Zero right now, there will be this 1 week of assessment of all the list of data points that are needed. Then an analysis based on machine learning will be conducted, and based on the placement of this patient in the distribution of the overall set of data, it will be assigned a specific treatment. So basically that's what I mentioned. And after the 3 or 4 months of treatment, depending on where the patient was clustered, then it will be assessed how this patient has been respond to that very specific treatment. The beauty of doing assessments like this is that you are able to mathematically calculate, basically, and include all aspects of the complexity of a patient's life, from what are the causes of the disease, what are the lifestyles of the patient, but also for the clinics, it allows them to stratify the way patients are treated. And it has a strong economic impact, and that has been one additional advantage of using these methods. This is another example, but basically during this period of time, from 2006 until now, there has been an improvement in the cost of efficiency of the patient, but also in the quality of medical care that patients are getting. We do everything for Zero, from health records digitalization all the way to the analytical part, to the analytics with the platform that we provide. These similar methods have been used for any other partnerships for other types of diseases. And basically, the goal is to make clinical knowledge useful without compromising patient privacy. And these are some of our clients and research partners, both in Europe and in the US. Thank you very much for your attention.
Afternoon, everyone. My name is Christian Capitini. I'm an assistant professor of pediatrics at UW Carbone Cancer Center, and I'm a pediatric oncologist. And I'm here to talk to you about CAR T cells and the new era of precision medicine for at least childhood leukemia. Here's my disclosures. So as you know, precision medicine has arrived in oncology in the form of cancer immunotherapy. What I'm showing here is the cover of Science magazine from 2013 when they announced cancer immunotherapy, depicting CAR T cells there as the breakthrough of the year. And we now have noted immune therapies being applied to basically every adult and pediatric cancer, both hematologic and solid tumor, that we treat. And we've noticed that these therapies can be used either alone or in combination with our standard therapies of chemotherapy, radiation, and surgery, and as well as small molecule inhibitors. But really, precision medicine in terms of immunotherapy started with monoclonal antibodies. And we now know that monoclonal antibodies can come in a variety of different flavors, You can conjugate different things to the antibody. You can use naked antibodies to attract immune cells to kill the tumor. And you can also inhibit critical growth pathways within tumor cells. And we've seen, at least in leukemia, a whole series of FDA approvals using monoclonal antibodies over the last 20 years, but particularly in the last couple of years, where we've seen expansion from chronic lymphocytic leukemia, CLL, to ALL with bispecific antibodies like blinatumomab, and then of recently inotuzumab and then gemtuzumab for AML. And there's many more that are in clinical trials. But today I'm going to talk to you about gene-modified T cells called CAR T cells, which use an antibody on its cell surface to recognize leukemia, but use the killing machinery of a T cell to eliminate the cancer. And how this process occurs is taking advantage of an observation in nature and how HIV causes illness. Because HIV is a retrovirus, and retroviruses, as you know, infect T cells. And once the virus is inside the T cell, it integrates in the genome and replicates, and then spreads and causes AIDS. When researchers figured out that they could remove the ability of the virus to replicate, they could take advantage of its ability to infect and substitute a code within it to make the cell express whatever it wants, in this case, the CAR product. So in this picture, you're looking at a viral vector that has the CAR DNA. That goes randomly into the genome, and then the cell then expresses the CAR, which is involved in antibody fragment attached to that killing machinery that then recognizes the tumor and kills it. And now there's a clinically established process where that we're able to do this right here in Madison. So American Family Children's Hospital was selected as a handful of sites around the country to offer CAR T-cell therapy now that it's FDA approved. And the way the process works is the patient, refractory or relapsed leukemia, would present to our hospital. We would collect T-cells from them in our apheresis center and cryopreserve or freeze them. Those frozen cells would be shipped to a manufacturing facility where the CAR T-cells would be challenged with these beads that stimulate T-cell proliferation, and the cells would be expanded from millions to billions over a period of about 2 weeks. At that point, using a lentivirus, which is related to retrovirus, that encodes the CAR gene product, would be used to transduce the T-cells and then make them express the CAR molecule on their cell surface. Then the cells undergo a week of additional quality assurance and quality control testing, are refrozen and shipped back to Madison, where they're thawed and infused into the patient. And basically, when the CAR is expressed, you get an scFv, which means a single chain variable fragment that can recognize a molecule on the leukemia called CD19. That molecule is also on our normal B cells, which are the cells that make antibodies and help us fight infection. But when it engages its target, it then gets a signal, a co-stimulatory signal and a kill signal to then kill the cell that expresses the target. And the results have been really dramatic. When Penn first published case reports using this CAR product, they saw that the cells expanded 1,000-fold in vivo, literally millions to billions. And what that meant is the patient became the bioreactor. We no longer have to grow the amount of cells you want to infuse in the patient. You just have to get to a minimal threshold, and then you let the patient's body become the incubator. And thus, these are, in fact, living drugs. But as a result of highly activating these cells, you see very unusual side effects that oncologists were not typically treating. And that included cytokine release syndrome, where virtually every cytokine that both T cells and cells in our host tissues can produce are produced at very massive levels. And then neurotoxicity, of which is a mechanism we don't understand, but could involve a variety of symptoms, ranging from confusion, seizures, to being completely made unconscious. And because of these very serious life-threatening side effects, the FDA required that this therapy be given in certified treatment centers under a REMS program, which is risk evaluation and mitigation strategy. And as I mentioned, Madison is lucky to be chosen to be one of the certified treatment centers. The patients go into remission that are sustained over many years without further therapy. All of the prior drugs that we had used were effective at inducing remissions, but they required the patient to get a bone marrow transplant to literally get a new immune system to keep them in a sustained remission and cure. If you just gave the prior drugs on their own, eventually the leukemia would relapse. But with this, we're seeing long-term remissions. You literally lose pounds of tumor. In the phase 1 studies in children with B-cell ALL, a 90% remission rate was reported, but that was at a single center. When we were then invited to participate in a multi-center international study with 26 sites around the world, the remission rate remained high at about 83%. The CAR T cells persist for a long time. We know at least you can detect them in the blood 12 months after infusion, but given that there's long-term survivors now over 5 years, we suspect that they stick around a lot longer because T cells naturally can form memory. The issue, though, is that because CD19 is on normal B cells, as long as the CAR Ts are in your body, you're getting rid of leukemia, but you're getting rid of your normal B cells, and thus you're dependent on intravenous immunoglobulin or antibody infusions to prevent infection for as long as the cells are in your body. And then lastly, some patients do relapse with CD19 negative cancer, which we had never seen before CAR T cell therapy. And just to put it in comparison with other FDA approvals, about 10 years ago, clofarabine was approved and had a complete response rate of 11.5%. And that's been followed with other chemotherapy drugs with pretty low complete response rates. And then with antibody therapies, I mentioned the bispecific antibody, which targets the same molecule, as well as a different molecule with inotuzumab CD22. The response rates range from 4 to 35%. With CAR T cell therapy, again, we've seen 83%, so remarkably higher than what we've seen before. And the durability is so far been surpassing all other therapies, here ranging from 5 to 8 months. We haven't even reached the median on the clinical trial yet. 12-month survival rate, if you give chemo, nobody lives 12 months. You need to get a bone marrow transplant to have a long-term survival. With the other antibody therapies, you do see a 30 to 40% 12-month survival rate, but you can double that with CAR T cell therapy to almost 80%. But there's also a big cost. So for these drugs, you have to give them repeatedly over either every day or every week or every few week dosing, and each dose, can cost you between $18,000 to $74,000, and it's unclear how many doses you're going to receive to go into remission. For CAR T cell, you get one dose, but that dose is $475,000. There are other CARs that are being tested in clinical trials or are being developed in laboratories around the United States, and we don't think childhood leukemia is going to be the only target. There's a pretty good chance that adult diffuse large B-cell lymphoma will be approved by the end of the year, and other non-Hodgkin lymphomas will also get approved probably early next year. But the real interest is in the solid tumor space for both adult and pediatric solid tumor cancers, and we really haven't seen any dramatic responses except on the case report level, but a lot of labs are putting a lot of work into that, and we should see that come to fruition in the next year or two. There are barriers to developing these very expensive cell therapies. First of all, for participation in clinical trials, this is just in general, not just for immunotherapy, but you do get resistance from insurance companies because they consider it experimental treatment, and even though it may have exciting results, they want you to treat with standard FDA-approved agents. There's also low reimbursement for clinicians to enroll patients on trials, so if you're not doing it at an academic center, there's very low incentive to enroll a patient in the trial if the reimbursement doesn't even cover the regulatory overhead. Some of the cell therapies in particular are at a very high cost, very expensive to develop. I've seen on average cell therapy trials ranging from $40,000 to $50,000 for just one infusion. NIH will fund, preclinical development, animal research, and they'll fund your clinical trial. But there's definitely this valley of death in between where your center has to validate that you can do the cell therapy, safely and with good quality control, but you can't get funding for that, and you have to rely on private foundations and philanthropy. And then immunotherapies in oncology have very high price points, as I mentioned, $18,000 per dose of a bispecific antibody versus $475,000 for a CAR T infusion. And then there's a lengthy review process, both when you want to get a grant, even if you're told today that you're funded, you don't see the money for 9 months. And then same thing with FDA approval. The requirements from the FDA can be onerous enough that it could take a long time to get your drug to market. So in summary today, I've told you that precision medicine comes in different types and different mechanisms, that all of them have shown an anti-tumor benefit. And I hope you've learned that CAR T cells are a genetically engineered immune cell, actually the first FDA-approved gene therapy that can recognize and eliminate leukemia cells with a very high potency, but at very high cost, both regulatory and financial. And we must find ways to overcome these clinical, financial, and regulatory barriers so that we can maximally deploy as many of these therapies as possible. Thank you.
Let me stay out. So I hope you all enjoyed those presentations. I think they're a good overview and shows you the diversity of the precision medicine activities that are going on in the state. I've asked our panelists to come join us up here on stage. And I'd like to encourage the audience, if you have questions, there are microphones. We have a few minutes left. We can take a couple questions. So please, maybe we'll start off with this group. I mean, one of the things that I've been noticing this morning already is we've had a lot of discussions about the triple aim, right? I mean, obviously reducing cost, improving quality, improving patient outcomes. How do you see precision medicine solutions influencing that?
Well, I mean, I think there's a lot of data in cancer care that cost to patients really can be devastating in terms of their care. And I think that we do need to think about this right convergence of efficacy, toxicity, and cost, and how we make sure that that we're considering that as we're counseling patients about the benefits and risks, but also how to explain that in terms that are manageable, are understandable. I think it's very difficult when you're faced with a very serious diagnosis to try to factor in cost and how that might impact your life too.
Other input, please?
Yeah, so for better or worse, pharmacogenetics, because it's been discussed for quite some time, we had for a long time, we always had the questions like, is pharmacogenetics actually cost effective? Because if you think about the test. And so there's a whole area which evolved as pharmacoeconomics and genetics. And the examples are coming out now. So there are clear examples where it's been shown that if you do pharmacogenetic testing, you adjust the dose, that for a number of different outcomes that this really saves money. Now, the thing in pharmacogenetics, what we can learn from this is it actually takes quite some time, right? I mean, from finding the gene which plays a role for a particular drug to develop the clinical evidence that this affects outcome, to the next step of like that the outcome affects really the cost. And I mean, one of the things what we can learn from that is that obviously collecting the information live, and then again, we're coming back to electronic medical records, collecting this data should really facilitate that. But I think one of the key things, what we learned, is that it will take time, and until we get to it and we accumulate this data, I think we shouldn't then just wait and say like, you know what, we don't want to do testing, because I mean, on the way as we generate this data, as we develop this and we get the evidence, patients will still benefit from this. So I think in the discussion of precision medicine, we shouldn't forget that really early on, like with any of the trials what you showed in cancer treatment, patients actually benefit right now from the treatment, and so it's a process, and we need to develop that.
Yeah, I agree, and I think even if we go through the process of patient stratifications, like in the zero case, it has been shown already for the Netherlands that it saves costs at the clinical level, at the patient level, at the insurance level.
Capitini?
Yeah, and I think also precision medicine companies need to become better at engaging insurance companies and explaining their technology, and also presenting a real financial argument on how it can save costs for the company, because they really look at bottom line in the short term and not really in the long term. And that's like that with most things in medicine, where they can see the value of a therapeutic that works immediately, but for a preventative treatment that may take 10 to 20 years to prevent something from not happening, it's hard for them to put quantification in that.
Well, unfortunately, we're at time for our session, but please join me in thanking our panelists today. And I just want to encourage everyone, we have a short 15-minute break, but please come back here, because we're hearing from Dr. Ru from Samsung Health and Fitness shortly. So thank you.